Our next speaker is Dr. Gabriel Corfas. Previously on the faculty at Harvard Medical School and Boston Children's Hospital, Dr. Corfas recently joined the faculty at the University of Michigan, where he directs the Kresge Hearing Research Institute and is a professor and associate chair for research in the Department of Otolaryngology. His research focuses on the molecular mechanisms involved in the development, function, and maintenance of the nervous system, as well as in using this knowledge to understand nervous system disorders and develop new therapies to treat them. Dr. Corfas.
Thank you, Michelle. Thank you, the organizers, and Michelle for inviting me. Today, I'll be talking about how postnatal experience changes the brain. And I would like to start by showing you some data from my long-lasting experiment. So when you're born, you have to learn many things: smiling, walking, driving, eating for yourself, having academic skills, motor skills, musical skills, social skills. And eventually, you end up being what at some point some of us consider a mature person. And this is a long way. And during this process, there is really a remarkable change, primarily visible in the nervous system. And neuroscientists have been interested in understanding how this happens and what is the basis for all this learning and changes in behavior. And 50 years ago, the amazing work of Hubel and Wiesel that led them to win the Nobel Prize showed that neurons in the nervous system are shaped by experience, and the connectivity in the brain changes with experience. And this was done using the visual system. They discovered how the projections from the eyes are eventually connecting in the visual cortex. They show that they're organized. There are specific columns in which each eye innervates the visual cortex. And eventually they show that during the early life, these neurons compete for innervation space in the cortex. And that if you see in a normal eye, it's very hard to do from here, but you can see that there are stripes very clear from each eye. And if you block one eye during this critical period in early development, the eye that sees covers everything. And this is completely permanent. And the eye that was not exposed at the right time loses the ability to drive visual experience. Now, this has driven the neuroscience community for the last 50 years to focus on how neurons are influenced by experience, how synapses are formed or lost, how projections and connections are made. Now, neurons do not live in the brain alone. They are surrounded by many other cells, particularly cells that are called glia. And one particular cell type that I would like to focus today is the oligodendrocyte, which are the cells that make the myelin in the central nervous system. Each one of the axons, or many of the axons in the central nervous system, are covered by some projections that are made by these oligodendrocytes that create the myelin sheath. And these are critical for regulating the speed at which nerve conduction happens in the nervous system. Now, this fatty structure in the brain generates what we call the white matter. And this is the microscopic way in which we have been looking at this for more than 100 years to look at the brain as the gray matter, where primarily cell bodies are, and the white matter, where the axons are. Now, for many years, like it was mentioned before, you look at the end of life, people were looking at myelin in postmortem samples. But now there are these new imaging techniques that allow for amazing qualitative and quantitative analysis of white matter. In the last decade, there's been increased understanding that white matter may be also influenced by experience. For example, there was an important paper that showed that children that practice piano from young age have different white matter structure than those that do not. And there was a pretty strong correlation between how much piano practice there is and how is the structure of particular areas in the white matter. Similarly, one very dramatic case of a link between white matter and experience came out when children that had been neglected in orphanages in Romania were analyzed. And these kids, which were basically deposited in these places and really completely neglected, were shown to have very dramatic behavioral and cognitive problems as they grow up. But also, some studies using MRI show that they have defects in white matter tracts in anterior parts of the brain. This is just one example from a paper looking at kids that were taken to England from Romania and showing that, for example, a specific tract called the uncinate fasciculus was defective structurally in these kids that had been neglected. Now, studies in humans are very good in creating correlations. We can see that neglect correlates with defects in behavior and defects in white matter, or practicing piano do that. But what we don't have with those studies is the power to analyze causality and mechanisms. So some of the main questions that remained at that time was whether the neglect is a cause. Is neglect and isolation changing white matter in the brain? What are the consequences of these myelin alterations on the behavior? Are they responsible for the behavioral alterations that were seen in children that had been neglected? And what are the mechanisms by which any experience changes the myelin in the brain? So we, as basic neuroscientists, decided to try to approach this question using mice. And today I will tell you what we have been learning about how rearing conditions change myelin in a particular part of the brain that is called prefrontal cortex, which is anterior and is very important for high brain functions, working memory, social interactions, and which has been shown to be involved in many neuropsychiatric disorders. So to start interrogating these questions, we decided just to take mice as soon as we separated them from the mother to avoid any issues of separation, stress caused by separation from the mother, which is when mice are 21 days old. And then we subjected to 3 different conditions. We kept them in isolation, 1 mouse in a single cage. We put them in a regular environment, which is how we usually keep mice, 4 mice in a single cage. And also, we gave them another environment, which is an enriched environment, which we thought it was going to be really changing something, which is a large cage with many mice and toys that we change every 2 days. There's been previous studies showing that enriched environment changes neurons, increases the generation of new neurons in the brain, and changes the connections. And then we ask whether these affects their behaviors, and we measure social behaviors. It's very simple. We look at how much a mouse is drawn to interact with another mouse that has never been seen versus an empty cage. And to test working memory, which is primarily from the prefrontal cortex function, we tested how well these mice can learn a pretty difficult task, which is to alternate in a T-maze between swimming to the right and then next swimming to the left. And this is pretty tough. It takes them 5 days to learn how to do this. And then we use structural and molecular techniques to look at what happens, the consequences. We use confocal microscopy to analyze in depth the morphology of oligodendrocytes, electron microscopy to look at the thickness of myelin, and PCR to measure gene expression. So the first experiment was isolate the mice between the day of weaning until they are adults at 65. And when we measure behavior, what we see is the isolated mice are not interested in interacting with other mice. But mice that are in regular environment or in enriched environment are pretty normal. And then what we saw is that mice that are in a rich environment or normal environment are able to learn this after a few days, this alternating behavior, but the mice that had been isolated could not learn it, suggesting that indeed isolation changes behavior in fashions that are very similar to what isolation in in an orphanage does. Then we looked at the oligodendrocytes, and we see that the cells are simpler, have less processes, and make less myelin. The myelin they make was also thinner, and the gene expression of these myelin proteins was reduced, suggesting that being isolated completely changed the program of myelination in the prefrontal cortex. Now, as I mentioned, human studies have been done with fMRI, and we wanted to know, by using these mice, can we validate what people are seeing in humans? So we sent the brains to Dr. Chu-Lien Liu in Duke, and he took these brains into to the MRI, and basically replicated what has been seen in humans, suggesting that what we are doing by isolating these mice for a few days replicates in very strong ways what happens to children that are neglected, and validated that the MRI studies on humans may be really seeing a consequence of the isolation. One of the concepts that came from studies of the visual system is that there are some critical periods during which experience can change neurons. So we wonder whether this was also true for the case of myelin. So we took mice and we tested whether isolation early on for just a few days would made a long-standing change in white matter and function, and whether this could be improved after by reintroducing animals to a social environment. And what we found is that indeed, just isolating the mice for 2 weeks generates a long-term change in behavior, oligodendrocyte morphology, myelin thickness, and myelin gene expression. But isolation later than P35, the day 35 of life, would not do it. And that bringing animals back into a social environment could not help in restoring normal myelination and normal behavior, suggesting that this early age, like in children, was producing a long-term change in their brain that had a dramatic impact on their behavior and cognitive function. So with this, we learned that juvenile social experience in some way regulates oligodendrocyte and myelin maturation, which is normal for normal prefrontal cortex function. And then we ask how, going to the mechanisms. And in that case, we decided to focus on a particular molecule signaling pathway that we believe could be important, which is the neuregulin ErbB receptor signaling pathway. And this is, I don't want to go much into detail, but these were molecules that we had already seen that when we modify their function, there is hypomyelination in the brain. So this is a complex system of molecules, neuregulin being a trophic factor that is produced by neurons, and acting on receptors that are expressed on the oligodendrocytes. These receptors are called the ErbB receptors. And when neuregulin activates the receptors, there is strong signaling to the nucleus that changes transcription, gene expression, and cell differentiation. So we generated genetically modified mice where at any time in life we can eliminate the receptors specifically in the oligodendrocytes. And then we tested whether eliminating this just one signaling pathway in oligodendrocytes before or after this critical period we have defined changes myelin and behavior in the mice. So what we did is we created mice and eliminated ErbB receptors in oligodendrocytes just at day 19, just the day before what we consider the critical period. And indeed, we get mice that have simpler myelin, thinner myelin, less gene expressions of myelin genes, and they have problems in social interactions as well as cognitive function. But if we eliminate the receptors just a few days later, what we found is that there are no changes. So first of all, what we found is that by eliminating this pathway and producing hypomyelination, we reproduce the same as if we would have neglected these mice, suggesting that just the ability to make myelin correctly is necessary for normal development. And remarkably, when we subject these mice to enriched environment, they still have the same problems, indicating that is the basic signaling and cellular processes that are occurring in the myelinating cells that are essential for making a normal circuit. So, of course, this gave us the notion that experience could be changing the function of this pathway in order to generate these specific alterations in myelin. So we wonder, is the isolation changing any of the molecules of this pathway? And using PCR, what we found is that isolation specifically changes only this molecule, neuregulin 1, in the prefrontal cortex, suggesting now we have the target. Now we know that isolation changes this gene. And by changing this gene, causes all these downstream events. So now we know that juvenile social isolation is necessary to maintain the normal levels. The juvenile experience is necessary to maintain the normal levels of neuregulin 1 expression, which is necessary to activate the receptor, which is necessary to induce the normal myelination of the prefrontal cortex, which is necessary for the prefrontal cortex function. And any of these steps, if it goes wrong, will create dysfunction. Now, you could imagine that this could be important in all the brain, and so we tested just the motor cortex, that is 1 millimeter posterior to the prefrontal cortex. And remarkably, it was perfectly normal, suggesting that isolation is very specific to this part of the brain, the prefrontal cortex. So, in conclusion, what we've learned is that social experience during the juvenile period has a profound and long-lasting influence on the formation of myelin in the prefrontal cortex by regulating the development of these cells, the oligodendrocytes, and that this is necessary for normal prefrontal cortex function. Also, that defects in oligodendrocyte maturation and myelination lead to alterations in adult social behaviors and cognitive function, similar to those caused by social isolation. So basically, based on this, we have come to the conclusion that in many cases, defects in children and adults that are being caused by social isolation could be mediated by specific changes in myelin in different parts of the brain. Now, since we published this paper, then a few other papers have come, and there's very much excitement about the notion that myelin, that we used to consider a very static component of the brain. We consider it mostly as the plastic around the cables that are producing our circuits and therefore even our conscious. Now it seems to be much more plastic. And there's been many cases where it's clear that activity of neurons regulates the process of myelin and development. Also, the work here by Michel also has shown this very elegantly. And even that the ability of oligodendrocytes to create new myelin is necessary for the learning of very basic skills, like walking on a complex wheel. This paper by Bill Richardson in England showed that if you eliminate the ability to make new myelin in adults, completely prevents animals from learning a very simple motor skill. So now, rather than being just the neurons that are subserving our learning, seems to be that myelin and oligodendrocytes are key for our ability to learn. Now, this has many implications. Of course, when you think about myelin disorders, the first one that comes to your mind would be multiple sclerosis, which is an inflammatory disease that destroys the myelin. And interestingly, many patients with multiple sclerosis have cognitive and behavioral problems. Now, with fMRI, it has become clear that many other individuals with disorders, neurological and neuropsychiatric disorders, have problems in myelin, including schizophrenia, bipolar, depression, autism, dyslexia, suggesting that many of the disorders that we used to think about, neuronal disorders, maybe also have an important component of myelin disorders, or maybe primarily a disorder of myelin. And interestingly, the genes that I mentioned, neuregulin, neuregulin and ErbB receptors, have been linked genetically to some of these disorders, suggesting that there is a unifying theme that these molecules and disorders of myelin may be contributing to important aspects of human mental health. And this is the people that did the work, and with many collaborators. And I want to leave you with the notion that oligodendrocytes may be a new, unappreciated component of the cellular machinery that our brains have in order to acquire new information, and that they are exquisitely sensitive to experience during the young years, and that is critical to provide the right environment and stimuli to allow for the proper development of myelin. Thank you.